across this nucleotide string. So that's what we're talking about in terms of genes. The question is then, what does a gene do? And of course it varies a little bit depending on what type of gene you're talking about. Some genes and their particular order of nucleotides are converted to another string, which is an RNA string. And that RNA string has a function by itself. Right? So that could be something like ribosomal RNA or tRNA or something like that. So that's a perfectly good function for a gene. It's a gene that encodes a very similar RNA sequence. What we typically think about as a gene is a gene that is transcribed into basically an identical RNA sequence, and that RNA sequence is then read off by the ribosome to produce a protein that has a very specific amino acid sequence. So we typically think of a gene as producing a protein that has an amino acid sequence order, and then that protein goes off and does something. Maybe it's an enzyme, maybe it's a structural protein, maybe it's a regulatory factor. Whatever it is, it goes off and does its business. How many genes do we have in humans? Roughly 20,000 or so. It kind of depends on what you call a gene and what you don't call a gene. And we know that a single kind of genomic region, a single locus, has in some cases the possibility of producing slightly different types of proteins due to things like alternative splicing and all this sorts of stuff. So this idea of a gene is a little bit fuzzy, but we can say it's on the order of about 20,000 different kind of discrete gene-like units. And other non-gene or DNA sequences in the genome maybe string all these genes together, right? So you have an enormously large genome, and a certain fraction of it encodes genes, but a lot of it is just kind of there, and we don't really know what all this stuff does. Sometimes people call this the dark matter of the genome, but there's a lot of nucleotide sequences whose order doesn't seem to matter a whole lot, but it just holds these genes together in these chromosomal strings. So that's kind of another aspect of the genomic organization. So we're going to go through a few topics today. A lot of these topics are basically connecting genes to proteins to steps in metabolic pathways. So the idea of what a gene does is kind of illustrated here. This is typically what we think a gene does. A gene produces an RNA, the RNA is translated into a protein, and the protein assembles or folds, whatever, and goes off and does what it's supposed to do. So here's some really glamorous pictures of various folded proteins. They may be multi-subunit, they may be single subunit, it varies from protein to protein, but ultimately the classic idea of a gene is that it produces one of these parts and then they assemble together. You get all these parts and the cell kind of runs itself, right? It has its metabolism and interconverts chemical to amongst one another's and so forth. So we typically think of genes as making parts, and if a part breaks, you have a disorder or a trait or something like that. Okay, so the first topic has to do with these so-called inborn errors of metabolism. This is one of the first observations made that would suggest that genes were connected in some way to biochemistry via enzymes that catalyze these pathway steps. So a classic example to illustrate this idea is a set of pathways involved in the metabolism of these aromatic amino acids, like phenylalanine, tyrosine, and so forth. And so what can you do with these? Well, you need these to make proteins, right? They're genuine amino acids that will be incorporated into proteins. They should be degraded if there's too much of them, so there's pathways for degradation here and here, and tyrosine can be metabolized further into things like melanin and certain other neurotransmitters and so on and so forth. So there's a lot of use to these types of amino acids. There's a lot of pathways involved, and it was known that there were defects in certain steps in these pathways, and this kind of led to the idea that these genes were producing enzymes which were controlling these steps. The first example here has to do with is it scratching? It has to do with a trait called alkaptonuria, which it just so happens has a block at this particular point in this pathway, and you don't have to memorize these pathways. They're concepts that we're illustrating about what genes do with respect to biochemistry. So there's a block here in this particular trait. It was observed that people with this blockage had urine that turned black in air, and this trait was seen in families, particularly very closely related families. So there was this notion that it was some sort of inherited disorder of metabolism. So that's one example, and that's one of the first ones that this guy Garrett looked at. And that's a long time ago, right? 120 some odd years ago. Other steps in this pathway which are blocked are this step here, PKU, and this step, one of these steps here leading to melanin. Okay, so the PKU is interesting because individuals with this disorder, that is 2 copies of a blockage of this particular step, will accumulate phenylalanine, and that turns out not to be good during development because the brain is very, very sensitive to imbalances in all these metabolites. So if you mess with amino acid metabolism and concentrations are too high here or maybe too low here, for instance, you cause brain development to not proceed correctly. And there are tests for the presence of too high of a level of phenylalanine in the bloodstream. These are usually performed on infants within a few days after birth. And one of the things that you could measure is whether phenylalanine levels are too high, right? And one, at least old way that people used to do this, I think I have it on the next slide, is called the Guthrie test. And what they would do is they would have a bacteria that required a lot of phenylalanine in order to grow. And so if the serum had lots of phenylalanine from some patient, the bacteria would grow. If the levels of this amino acid were normal, the bacteria wouldn't grow. There may be more up-to-date tests for this these days, but every infant gets tested. And part of the reason every infant gets tested is because the allele frequency is high. If the allele frequency is exceedingly low, you tend not to worry about it because chances are everybody's okay, right? But for some disorders like PKU, you can fix it if you know it, and the allele frequency is sufficiently high that it's worth to bother to do tests early on. And fixing it, in this case, involves basically trying to restrict phenylalanine in the diet. Okay, so the idea here is similar to the idea we saw with those pea plant flowers and that little pathway we saw there. That is, you have a gene, it encodes an enzyme, the enzyme accomplishes some step in a metabolic pathway. In this case, we have a step here, and some step over here. Is it crackling like anything? Yeah, somebody resets the gain on this thing sometimes, and then it's messed up, and I have to call them and say, hey, can you turn it down? So sorry about that. Okay, so yeah, you still hear me? Is it better? I'll try this. Okay. So anyway, that's kind of point one. This idea that genes affect these metabolic steps because they produce individual enzymes that catalyze those reactions. So another set of old experiments that reinforces this same idea and gives rise to a nice type of genetic problem is this Beadle and Tatum one gene, one enzyme idea. And with this, you can map pathways, biosynthetic pathways, for instance. So we're going to look and see what they did and how this works and how you could get a set of data and create a pathway. Okay, so the organism they used is a fungus called Neurospora, and it comes in 2 mating types, big A and small a. So like most things on the planet, there's 2 mating types. Big A can grow in some haploid state just fine, and the small A mating type can also grow in a haploid state just fine. But once in a while, if they encounter one another, there will be mating between big A and small a mating types to form a diploid. That diploid will then undergo meiosis to generate haploid spores all over again, and then they will grow in the haploid form. So there's a life cycle involving a haploid stage and then this fertilization and meiotic stage. So some characteristics about the growth of these things: they can grow, if they're normal, on minimal media. Minimal media just contains some sugar, some nitrogen, maybe a few essential vitamins, but otherwise not too much, maybe some salts. So a cell should be able to grow on minimal media because it can produce all these biosynthetic enzymes, and it can make its own amino acids, its own nucleotides, all this type of stuff. So most cells should be able to grow on minimal media. Some cells can't. And one of the reasons they might not be able to grow on minimal media is if they have a defect in the biosynthesis of one of the important components, like an amino acid or like a nucleotide. In that case, you'd have to supplement the media with a bunch of other stuff in order to allow them to grow. You might supplement them with everything. That would be called complete media, and it would give them everything that they need. They wouldn't have to bother with making anything. Or you could take minimal media and you could supplement it with just one particular compound that you knew that cell was defective for, in which case it would then grow on minimal media plus that compound. So this is some characteristics, and we're going to use this as a screen to try to identify these biosynthetic mutants. And we've got to make the mutants. How do we make them? In this case, we irradiate the Neurospora with X-rays. The X-ray is obviously now a known genotoxin, but you could ask the question, many years ago, is X-ray damage permanent, or is it just something that kind of breaks apart proteins and everything else in the cell and is a temporary? And the answer is, well, X-rays induce genetic damage because the damage that you produce is heritable. So generation after generation after generation, you find the same defect in the offspring of cells that were irradiated. That would suggest it's a permanent genetic damage and not some temporary transient damage to the cell. So the question is, what about, is it in germ cells? And for an organism like Neurospora, the whole thing is basically a germ cell, right? The definition of germ cell doesn't apply in the same way here that it would in the higher mammalian organisms. Okay, so here's an example of the life cycle of Neurospora. There's a lot here. You don't have to worry about all the details. I just want to highlight again this idea of haploid to diploid to meiosis and back to haploid. So we have a type of cell here, which is the big A mating type, a type of cell here, which is the small a mating type, and they grow perfectly well. Here they're growing, and here they're growing. And this is kind of interesting because in order to be good at with a haploid genome, all the genes have to be good, right? You can't have any severe defects. So this kind of reveals anything that's bad. So going through this haploid life cycle almost makes sure that this cell is a good quality genotype and can do everything it needs to do. Anyway, so we have the fusion of the big A and small a cell types. That makes the nucleus 2N. It's now diploid. Then we go through the same type of meiotic cell division we talked about before, where you have replication, you've got recombination, you've got a couple cell divisions, and we end up with haploid spores or haploid offspring. It just turns out in this particular organism, another cell division occurs so that actually you end up with a total of 8, but that's not that important for us. The point for us basically is you go from haploid to diploid, you undergo meiosis, and then it just so happens you divide again. So you go through this haploid to diploid cycle. So we're going to make use of this life cycle in this beetle and Tatum experiment to try to identify mutants in biosynthetic pathways. Okay, so a summary of this is illustrated or is written here for you to read, and then a picture of it is in the next slide here from the book. But the picture is so small, I kind of wrote out what the steps are in this process. So the first thing is take a line of neurospora and irradiate them, that generates permanent genetic damage. So now the cells are probably defective. Many of them may be defective, so they can't grow. But you get around this with a trick. You make these irradiated cells to the opposite mating type, which is wild type. Now they're diploid. Diploid masks these mutants that you produced by irradiation in the haploid strain, right? So that's pretty slick. Now the cells are okay, and they're okay long enough that they can undergo meiosis and sporulate and produce those that tetrad of haploid cells. So the fact of mating with wild type overcomes, just as it overcomes in organisms like ourselves, any defective alleles that were created by irradiation. We allow them to sporulate. This regenerates haploid organisms. If they have a defect, some of them may not be able to grow, and that's kind of what we're going to screen for in the next step. So we can select out single cells. You can actually dissect this type of cell. You can select out single cells, and you can put it in a test tube. And the first thing you do is you put it in a test tube that has complete media. So even if the cell has a defect, it still might be able to grow up, right? Even if it had a biosynthetic defect, it still might be able to grow up because you're supplying it with all the nutrients. So the first step is just to get the thing rolling, just to get them growing. So mutants and biosynthetic genes will grow, unmutated cells will grow, and so forth. So you can imagine that after irradiation, you've got multiple things that might have happened. Some cells get no hit whatsoever, right? They will grow because there was no damage. Some cells might get a hit in a biosynthetic gene, something that makes leucine or valine or something like that. They would be able to grow because you're supplying it with everything. But some cells would get a hit in something really important, like a DNA polymerase or some other essential gene. Those cells won't grow. Okay, so you won't see growth even in complete media if it just so happened you got a hit in something ultra important like that. So we're kind of parsing out the types of mutants already that we're getting when we do this first step of growth in complete media. Okay, so we then take, after they've grown in complete media, we take a little bit and we transfer it to minimal media, and we ask the question, can it also grow in minimal media? And if it can, there was obviously no defect in some biosynthetic pathway. But for some of them, you put them in minimal media and they can't grow. And that would suggest that they grew in complete, they didn't grow in minimal, therefore there's something present in complete media that provided some essential component that they were now defective in producing. So we look for cells which we screen in this manner, growth in complete, failure to growth in minimal, and then we add back things and ask, well, what was it? What was it in complete media that allowed the cell to grow? So that's basically the idea of what we're doing here. The picture shows you the same thing, but it's a little bit odd picture. You irradiate, you cross with the opposite mating type, which is wild type. You dissect out the single cells. You hope that some of those have a defect in some biosynthetic pathway. You grow them on complete media. This is supposed to show that they're growing somehow, that they're growing here. Okay, and then for each one of these, you take a little bit, you transfer it to minimal media, and somehow this is supposed to show that one of them didn't grow. That means there was something in here that allowed growth, but when you asked it to make that something, it couldn't grow. So now you ask the question, well, what is it in complete media that allowed growth and disallowed growth in minimal media? And so you start to add things back. Could be a nucleotide, could be a vitamin, could be amino acid, could be lots of things. So you add things back and you ask, what allows growth when I add this back? If you add back all the amino acids, in this particular example, you see growth again. And then you ask the question, well, which amino acid? And that's the experiment at the bottom. The idea is here that in this case you added tryptophan back individually to minimal media. The cell now grows. Therefore, the mutant cell that we've isolated in this process had a defect in a gene that helped control this tryptophan biosynthetic pathway. So that's the logic of that. And you might get lots and lots of these tryptophan mutants, and they might be in different genes, right? And you could gather that information and you could start to organize the pathway. Yeah, question? not sure I understand your question. Can you ask it again? So you're kind of, I mean, if they're not defective, then you would see that maybe they would grow as well in minimal media, right? But you're not interested in that. Does that make sense? I mean, you have basically some cells that are defective in some biosynthetic pathway here, and some that are completely okay. But when you grow them in minimal media, you're kind of moving away this group that's okay for synthesis of everything, and you're just kind of isolating or identifying that category which is defective because it can't grow in minimal media. If I understand your question. Okay, so anyway, like I said, you might get lots of mutants, all of which require tryptophan, and they don't have to necessarily be in the same gene. They could be in any gene leading to the final tryptophan product. So we can take all these mutants, we can look at their growth characteristics, and we can order a metabolic pathway. And that's really where we're leading or heading in this type of problem. So a typical set of data that you might see would be one of these growth charts or growth tables. And in these, we designate growth usually as plus it grows, minus it doesn't grow. So that's pretty obvious. And usually what we have are a set of mutants that will fail to grow in minimal media, okay, because they have a biosynthetic defect. If you provide them with the endpoint of that pathway, whatever amino acid they failed to produce, they will all grow. So here's minimal media added with nothing. None of the mutants can grow in minimal media, all right, because they're defective. If you add the very last compound, which is not Pearson, Benjamin, Cummings, but methionine, the last one, they all can grow. So they were defective in the production of methionine. Okay, and the question is, are they all in the same gene, all these mutants, or different genes, and so forth? And so that's kind of how this is set up. We have a set of mutants that are defective for methionine biosynthesis. They can't grow in minimal media. They can grow if you supply them with methionine. And the other thing we have to know is a set of compounds which we already know are in this methionine biosynthetic pathway. So this is kind of pure biochemistry and pathway tracing type of stuff. So let's suppose we know that the pathway involves a series of chemical precursors, like O-acetylhomoserine, cystathionine, homocysteine, finally leading up to methionine. Maybe it's in this order, maybe it's not. It just depends how you portray the table, but we know that there's a series of compounds that are going to be interconverted whose end product is finally methionine. Okay? And we know that these have to be converted amongst one another by the products of various genes in this methionine biosynthetic pathway. So let's look at the MET8 strain. It's mutant in some gene, which is designated here as MET8. What are its growth properties? Well, it can't grow in minimal media because it's defective for methionine biosynthesis, but it can grow if you add methionine. But the important thing to note also is that when you add all these precursors, these intermediates in this pathway, it can't grow with any of these either. That suggests the block in this MET8 mutant is at the very, very end of the pathway. The only way you can overcome the block is by adding that very last product. Okay, so that's the logic of these things. The answer is already shown to us down here. The idea is methionine is at the end. There's a gene encoded by MET8 that catalyzes this last conversion from something to methionine. Okay? If this is blocked, it doesn't matter whether you add anything before it. It'll all kind of build up at this point and stop. The only way you can get this strain to grow is by adding the very last product. So this logic seems easy initially, but it's also easy to get confused by. Let's see a question. Okay, so in this particular case, we're only looking at the MET8 strain by itself. There's only one mutation in this MET8 strain. that one mutation is in the MET8 gene right here. Everything else is perfectly good. Does that make sense? Right, so this is an assumption that you have to make, or you have to test, or you have to figure out. If during, and I didn't say this, but if during this process of irradiation, there's several things that could happen. One is you use too low dose, in which case most of your cells are completely normal. So you're just going to screen and screen and screen and screen, and nothing ever happens because you use too little. And the other end of that is you use too much. Too much means a lot of them die because it's dangerous, and you get a lot of them with multiple mutations. If you get cells with a lot of multiple mutations, it will really confuse you later because you will have to then go back and say, well, is it due to this? Is it due to this? Is it due to this? All these things would be questions you'd have to answer. So when they do these experiments, they have already kind of calibrated this as best they can to try to get one mutation approximately per cell, to the extent that that's possible. Yeah, but you're right. If there are multiple other mutations in this MET8 strain, there would always be this question, well, maybe it's that gene that's causing it, maybe it's not this. So we're going to say that they've done all this work, and they have provided the answer, and it's right, and they know that it's MET8. You could make the prediction it's MET8, then what you could do is take a normal strain, and you could deliberately change only the MET8 gene and see whether it acted the same. That would kind of prove that, in fact, it was this mutation. So there's a lot more behind these than just the table that you see. Okay, so in this strain, there's only one mutation in MET8. Everything else is good. The pathway is blocked to this point. No matter whether you try to add any of these, you can't jump across this step because MET8 is mutated only if you add methionine. So we position MET8 at a particular place in the pathway. MET2, we don't have to go through them all, but let's look at a few. MET2 is a different mutation in this methionine biosynthetic pathway, and you know it's different from MET8 because the growth characteristics are different. In this case, for MET2, if you add the very last compound, you're good. That makes sense. If you add homocysteine, you're good. But if you add these other two, you're bad. And you try to organize the logic of this in your head and come up with a pathway. And again, they've given us the answer here. But let's suppose the MET2 defective allele encodes an enzyme that catalyzes this step right here. Something that produces homocysteine, which then can go on to produce methionine. If that were the case, then you could see that if you added this or this, you'd be blocked right here and you'd be defective. On the other hand, if the block is here and you add anything after it, like homocysteine to methionine, you're good. Right? So that explains why this strain can grow on homocysteine and it can grow on methionine. Okay, so from this kind of growth table, you can start to develop a pathway of intermediates leading to synthesis of methionine. Let's do, what's the last one? MET5. Let's do MET5 because it's at the other end. Okay? So MET5, minimal medium alone doesn't grow. You add any of these, this one, this one, this one, this one, it can grow. That means the block was very early, and you add any of those later compounds, they can all still flow to the final end product, which is methionine. So in the diagram, we have the MET5 strain here. It's a mutation in this MET5 gene. It causes a block in this metabolic transformation right here. And if you add anything after the block, this one, this one, this one, this one, you get growth. Okay? So in these types of problems, which are a pretty common problem, you're basically shown usually a set of data about the growth characteristics of various mutants, and you're then asked to develop a biochemical pathway where you have a series of compounds converted from one thing to another, and you're asked to then place the locations of the mutants or these strains along this biochemical pathway. So some variants of this will be, what if there's a branch? Not all pathways are linear. If there's a branch, I mean, that's a possibility, and that will confuse the table that you see, right? It'll make this more complicated. In fact, I think I remember in the book there's a problem involving some branches that are just mind-boggling. But nonetheless, a branch is a reasonable thing, and it will really confuse us. This is a pretty straightforward type of linear path. The other thing that I find people do is they reverse the order. They get the pathway right, but they switch the endpoints. Okay, so you want to be aware of that possible danger. And I find also that these problems seem simple when you look at it or someone talks to you about it, but when you see one of these things just cold, your mind goes all fuzzy, okay? And you're wondering, well, what's a pluses? And you get confused. So, it's try to get the fundamental logic of this, because if the table is rearranged, anything looks a little different. You have to be able to understand the fundamental logic of what's going on so that you can kind of recapitulate the pathway as you're supposed to. Okay, so the idea, one gene, one enzyme, right? Each gene is encoding something that is catalyzing a particular metabolic step. This idea is super popular in genetics. It holds true for lots of things. It's a real Mendelian type of an idea. You have a gene that controls a particular feature or phenotype. This is just a partial table of all sorts of things that are defective in humans with respect to genetic disorders, where this basic idea of one gene, one enzyme has held true, right? And you can look at this online Mendelian inheritance in man, where people have tabulated as much as they possibly know about various genetic defects in this gene and that gene and the other gene, and what output they have in terms of symptoms and phenotypes and so on and so forth. So basically we're parts, right? We're composed of these parts. The genes are parts. These gene parts make protein parts, and the protein parts make organisms. And if the gene part breaks, the protein part breaks, and you have some consequence. Not that much different from a car, right? If something breaks on a car, it's a very specific thing. You have to repair that. Bodies are made of these kind of parts as well. This is different from the idea of complex genetic diseases, which are due to these quantitative, additive effects of lots of genes adding up and producing an output in some phenotypic curve. It's a little bit different concept than that we just finished with. Okay, topic 3, which is a miscellaneous set of stuff, but basically it's the idea that you can actually see the output of a gene in certain situations. And we're going to look at an example involving the hemoglobin gene, and we can actually detect what the genotype is using some biochemical approach. And this will tell us something about the nature of alleles as well, which will be important. So the title is Detecting the Presence of an Allele Using a Biochemical Approach. And the model is the sickle cell allele, which is a mutation in the beta polypeptide of the hemoglobin protein. Hemoglobin, its function is to carry oxygen, and it's an alpha-2, beta-2 tetramer. It requires 2 separate genes, an alpha gene and a beta gene, and it's known that there are a variety of alleles that affect the function of these alpha and beta proteins, which have various consequences. So the one we're going to look at here has to do with this sickle cell allele. The distribution of this varies. You can look up these maps showing the allele frequency around the planet. It's kind of interesting. But the change in terms of the sequence for us is shown here, is we have a normal beta polypeptide, and we're just showing the N-terminus of it. And so we're given amino acids 1, 2, 3, 4, 5, 6, 7. And we're saying that at position 6, we should have a glutamate, right, which will be negatively charged. And in the sickle cell allelic variant, there has been a nucleotide sequence change in this beta hemoglobin gene, which causes the substitution of a valine for the glutamate. Okay? And as a consequence of that, this protein acts differently than the normal protein, and if you've got 2 copies of it, it can cause the blood cells to deform in this kind of characteristic sickle cell shape. The idea in part based on the distribution of alleles around the world, is that if you're heterozygous, it may have some advantage in terms of resistance to malaria. That's what they say. Okay. So anyway, here we have homozygous normal beta protein. Here we have homozygous sickle cell allele. So we want to look at the kind of biochemistry of the production of these alleles in different individuals. So we can use this technique called electrophoresis. There's all kinds of varieties of electrophoresis. This particular one asks whether the protein is more positively charged or negatively charged on the surface, and based on that, it will move in different directions on a gel. In this gel, so we have a cartoon picture of the gel. We have plus applied here, minus applied here, and what we're going to do is take blood from an individual and maybe purify the hemoglobin. Probably hemoglobin is going to be very abundant, so that's not going to be a big problem. And we load the hemoglobin protein at some point here, and what we're seeing in this particular kind of cartoon is that having 2 copies of the normal beta protein cause those proteins to migrate over to this particular position. An individual that is heterozygous for one normal allele and one sickle cell allele produces 2 different types of proteins which differ based on this sense or this substitution here. And you can, in this assay, you can actually detect that difference based on the charge of the overall protein in this electrophoresis technique. And an individual that's heterozygous produces 50% of the beta protein off the sickle cell allele, which is here, and 50% of the normal hemoglobin off the other allele contained in that person. So you see 2 distinct bands reflecting the production of equal amounts of protein off these 2 different alleles. And in the picture, they've tried to show you that you get 100%, you get 50%, and 50%. That's what they're depicting with the intensity. And then finally, a person that is homozygous for the sickle cell will not show any protein migrating at the position of this normal protein, but all 100% will migrate over here. So this is kind of a neat assay. It provides a window into the genetics of the individual that's being tested. You're looking at proteins, but from proteins, you can make predictions about what the allele content in that individual is. So that's pretty nice. The reason we use hemoglobin is in part because taking blood is fairly non-invasive, right? If you were talking about alleles of some protein that were, let's say, expressed in the brain, I mean, it's not going to happen, right? You can't open that up and take something. But blood, that's why blood disorders are studied so frequently, because the access to material is much easier than some sort of internal organ. And these days, you might not do this. This is a kind of older, laborious technique. These days, what you do will be some sort of direct sequencing, maybe PCR. You know what the sequence is, you know exactly the nucleotide. You will use some molecular approach to make these determinations, rather than collecting blood, isolating proteins, and running it on a gel in this kind of laborious fashion. Okay, so kind of a neat assay that lets us see the genetics behind the individuals that are tested. So this slide is to re-emphasize the idea that each of these genes comes in lots and lots of allelic forms. This is the general rule across populations of genomes. So what we have here is a depiction of the alpha chain of hemoglobin on top and the beta chain of hemoglobin on the bottom. And you have at the top of each of the A and B, you have the normal amino acid sequence at selected residues along the length of the protein. So for alpha chain, you have leucine at 2, lysine at 16, so on and so forth. Okay? That's the kind of wild type expected version. And then below, you have a bunch of variants that have been identified through various techniques where people probably came into a clinic for various reasons, and ultimately it was discovered that there were substitutions at various positions along the length of this alpha chain. So it's an idea that there's lots of possible allelic variants available in a population, and they exist. They're potential alternatives for being homozygous wild type for this alpha chain. And the same is true for the beta chain. We saw one, which was this change at position 6, but there's a number of possible changes that can occur at other residues. And this is by no means complete, right? You can have lots and lots and lots of possible nucleotide changes along the length of a gene that may have an effect, may not have an effect, and so forth. So this is obviously going to be a very small subset of possible changes, those which caused amino acid substitutions, those which probably had some clinical effect, otherwise they might be never seen, right? If you had a substitution, let's say valine to isoleucine, where the character of the amino acid is similar, you might never observe that. It would never become clinically obvious. So probably most of these arose because there was some clinical consequence. But nowadays with PCR and sequencing, you could sequence 1,000 people or 1,000,000 people or whatever, and you could observe all the possible sequence changes or allelic forms that would occur across the entire gene. And the diversity is immense. Okay? So that's what we're kind of getting at in these real glaring examples where you see various possible allelic forms where there have been a major substitution of one amino acid for another. Good. Just a few more things and we're done. I want to spend a little bit of time talking about alleles of genes and where these changes in nucleotides may occur when you talk about one allele versus another, and so forth. So before I mentioned that, let's imagine you have a 100 nucleotide string, and the sequence of this string of nucleotides is exact. Okay? At any given position, for whatever the nucleotide is at that position, you could change it to 3 possible other nucleotides. So if it's G, you can change it to something else, right? CAT, right? And so forth. So even for something as small as 100 nucleotides, you've got an immense number of possible variations, and that would be just considering one change only in that thing. You might have multiple changes. So you have an incredible variety of possible nucleotide sequences, even when you limit yourself to just 100 at a stretch, which is a meaningless number with respect to gene sizes. It's very small. So there's lots of places where you could have sequence change that might cause some effect. And so I want to go through kind of point by point or region by region where you might have a change in the nucleotide sequence and whether that would cause some phenotypic change, some effect on the organism. So point 1, if you have a nucleotide sequence change in the protein coding region, and if it occurs at a position, so it's going to occur at a position that affects an amino acid sequence because it's in the region of the DNA that will encode a protein, you can have a couple of things happen. One is you could change the amino acid sequence from one thing to another thing. That's the example we just saw with these hemoglobin alleles, right? That's possible. Another possibility is the nucleotide sequence change would result in the termination of the protein altogether. The ribosome would fall off. Okay, so these are some things that could happen for a nucleotide sequence change that occurred in the protein coding region. Would they matter? Well, for termination, if termination occurred one codon upstream of the normal termination codon, probably no big deal, right? You're still making the entire protein, pretty much, so probably you're okay. But if a nucleotide change resulted in a termination event just downstream of the first methionine, then you don't make the rest of the protein, and so that's a big defect. That's kind of null. You're defective for production of anything off of that allele. So will the termination codon matter? Most likely yes, but it's partly position dependent. What about a change in amino acid? Yeah, usually this has an effect, particularly if you change amino acids that are of different types. Acidic, switch to basic, polar, switch to something bulky like proline, aromatic eliminated for some small hydrophobic, something like this. If the change or the character of the amino acid varies quite a bit due to some sequence change, then it's very likely, or at least more probable, that the protein that's produced will have some defect compared to the wild type protein. Point 2, let's suppose it's in a protein coding region at the position 3 of the codon. You know from that genetic table that you need 3 nucleotides to specify an amino acid. If a nucleotide sequence change, some allelic form occurs in that 3rd position, then the substitution might be synonymous and there's no obvious effect. You change valine to valine, who cares, right? It's an allele, technically, because there's a sequence change, but there's probably nothing visible ever going to happen if you change valine to valine. It's itself all over it. Okay, what about in an intron? So you know in eukaryotes, genes are divided into exons and introns, right? So the genes are really big, but the part that encodes the protein is pretty small, and it's separated or dispersed across that region. So you have a little bit here, a little bit here, a little bit here, a little bit here. Those are exons, they code the protein. You put them together by splicing, and then that makes the amino acid sequence of the protein. And the sequences that separate the exons are called introns, and those sequences are free to diverge pretty readily in sequence. So if we make a sequence change in an intronic region, that would be an allelic form of some other sequence of the same type that didn't have that change, but would it have an effect? Probably not. So in all probability, if you have a sequence change in allelic form occurring in the intron, the effect is usually minimal. If there were something encoded in the intron we didn't know about, then maybe it would matter. If the mutation hit a splice site that affected splicing, maybe it would matter, right? But there's a big sequence in there that you can hit things all over and it just doesn't seem to matter. So the general rule is if it's in an intron, effect is probably minimal. Promoter region. You have a sequence change between 2 genes making allelic forms, and the sequence change occurs in the regulatory region, just upstream of the body of the gene. Will it matter? Again, it kind of depends. If that sequence change affects a transcription factor binding site that regulates that gene, then maybe you're going to have some consequence. There could be different outputs from those 2 alleles. On the other hand, sometimes sequence changes in the promoter don't have any effect. It doesn't hit anything important, and so you're okay. So it kind of depends when you think about sequence changes in the promoter region. And the last one I have here is, what about regions between adjacent genes? So you have a gene here, you have a gene here, and you have all this kind of dark matter of DNA sequence connecting those things together. If you have sequence changes in here, they are technically allelic differences from one genome to another genome to another genome. Will they matter? Usually not. Right? Usually that stuff is just thought to be connecting material. It's possible something is in there that you didn't know about, but more often than not, you can change sequences in some of those regions, and even though they're technically allelic differences between individuals, they may not have some real clear phenotypic consequence. Okay, and then the last comment here, the type of mutant alleles we've looked at so far typically are in this category, right? Typically, we've looked at these dominant and recessive alleles where you have one sequence which produces a good enzyme that does something. You have another allele, the recessive one, which generally is defective in some way. Maybe because it terminates translation and you don't get the protein. Maybe because there's a mutation that inactivates the protein and it can't perform its enzymatic function. So typically, dominance and recessiveness is a consequence of something like this allelic change in category 1. Okay, and then loss of function, gain of function. In the typical Mendelian genetic examples we've looked at, the recessive is often a loss of function, just like I mentioned. You're creating a protein that's broken and doesn't work, or you fail to synthesize it in the first place. So if the mutation stops the protein from being produced or it stops the activity, the resulting protein has no activity. That's loss of function. So you've got one allele here, one allele here. This is a good one, makes a protein, it's active. The other allele is recessive. Often that's because it's defective and you get no protein activity output for some reason. Okay? That's a loss of function. You lose the function of the recessive allele. In diploid organisms, we always argue that the rationale why diploidy is important is because if you have one allele that displays this loss of function, the other allele compensates and gives you a good protein. It's only if you're homozygous recessive that do you then fail to produce any active protein from that particular locus. And for most genes, producing 50% off one good allele is okay, right? That's why dominance and recessiveness works. There are a few genes that are termed haploinsufficient, which means haplo, one copy, not enough, insufficient, right? There's a few examples of this where if you just have one good copy of a gene and the other one is defective, having only one good copy isn't enough and there will be some phenotypic consequence. But these are kind of unusual special examples. Typically, we imagine that having 50% production of one good allele masks or overcomes a loss of function allele on the other chromosome. Okay, so that's fine. And I think the book also discusses gain of function. So gain of function is a little bit different. In gain of function, you have a mutation or an allele that's formed by some nucleotide sequence change, for instance, and you get something different out of it. The allele is acting differently. So some examples would be, maybe you have a substitution of one amino acid for another amino acid near the active site. So now you're still producing the enzyme, but its specificity is a little different. So that would be something you gain, right? As long as you had that allele, you would gain that particular new substrate specificity. Another example would be if you had a mutation in the promoter or regulatory region that caused that gene to be expressed in a different way, too much, or in different tissues or something like that, that would be gain of function. If you had that sequence, you'd always observe that kind of new expression profile or new pattern of expression. So those are examples of gain of function. And the way you might think about it is that imagine you have a gain of function allele along with the normal wild type allele, and I just indicate it by an asterisk to kind of indicate that you get something out of this. It's not just missing, you get something. So this would be, in a sense, kind of dominant. So you get the output of both this one and this one. And that might cause some phenotypic effect depending on what the gain of function would be. Okay, good. I think we're almost done. Let's see, conclusion, just a few simple things to conclude. One is classical Mendelian genetics describes this pretty simple one gene, one enzyme idea where you map genes to particular points in these metabolic pathways. That's all fine and good, kind of exemplified by this metabolism of aromatic amino acids. That's fine. But these days, people think of the cell as this super complicated interconvertible set of reactions involving a huge number of chemical compounds, which rely on an enormous number of enzymes encoded by lots and lots of genes. So the real biological system is more like this, and even this is simple because it's a bacterial cell, but all these steps have to be encoded by genes, and they're all going to come in different allelic forms that are going to be active, more active, less active, certain substrate specificities, so on and so forth. So the character of even a bacterial cell is going to depend on all the alleles it happens to have in its genome. And you can imagine that if you have a defect at any given step, you're going to cause kind of a change or in the dynamic or the flow of all these compounds throughout the cell, right? So you can imagine lots of kind of pleiotropic effects as a consequence of mutations in various steps here. So something that happens here can have an effect over here and vice versa. Now, we don't really know how to analyze these problems. It's exceedingly complicated. We can do one gene, two gene, three gene, a few things like this. But after a while, there starts to become so many variables that you have to input and take account of that it becomes very, very difficult. But we can see that this is kind of a problem if you kind of look down at it. Okay, good. So last thing, just a few quirky examples of genetics in the news. If you're a genetics student, you should pay attention to how the popular media presents genetic type subjects. So I have some examples here just for fun before we go. So covers of Time. Here's the first one. They're not in any particular order, but roughly chronological. So here we have what kind of an Adam and Eve type of depiction of two mating types encircled by DNA double helices that are kind of winding up into trees in some sort of nature thing. And The title is Solving the Mysteries of DNA, and it's supposed to be a celebration, 50th anniversary celebration of the Watson and Crick discovery of DNA double helix. So here's one. Next one, genetics, the future is now. Yeah, so the future is never now, okay? The future is the future, but they say the future is now. And I don't know, after class, whether you go back to your apartment or your home and you stretch out like this with just, you feel like this after genetics, I don't know, but this guy feels like this after genetics. Is there anything wrong with this picture? I was asked this, some of you may know already, but if so focus on the double helix that they put in his body, or her body, whatever it is. It's left-handed, okay, so they messed up. This one is going around in the correct polarity, but this one is going oppositely. This is a pretty common mistake. There's even somebody who used to have a website where he would tally all these examples where people used accidental left-handed helix when it should have been a right-handed helix. And you see sculptures, right, where they did it wrong. They messed it up. This is probably, this is pretty forgivable. He probably, the person probably has an image and it's reversible. You can put it in one way or the other way and didn't think about it. And it's good, you know, that they got this right. The worst case of this was the issue of Science that reported the human genome sequence. They got it wrong on that most important issue. They reversed it there too. Okay, another one. This is Shaping Life in the Lab, the boom in genetic engineering. And Herbert Boyer was one of the founders of Genentech, which is a big biotech company. And they put his face here. Not the most flattering picture, perhaps. He's got all these things coming out. I think these are supposed to represent DNA or nucleic acids in some way, but I don't know. I'd ask for a redo, I think. Here's another one, the new genetics, man into Superman. Again, I don't know, man into Superman. At least here, they've gotten away from the double helix. They haven't made a mistake, right? They've made it two-dimensional, so they're safe. But the format is pretty similar to the first one. Okay, a couple more. Dolly the sheep, will there ever be another you? This was a big media thing when it was realized that you could clone animals, and Dolly the sheep was one of the first. Right? You can make an identical copy of an adult organism by taking the nuclear DNA, the diploid nuclear DNA, out of an adult and putting it in an oocyte that you've removed the nucleus from, and you can cause that diploid DNA to program development and do exactly the same organism. That's why there's that website I showed earlier. Was it my pet? Something like that. You know that one. My friend again. Yeah. My friend again. All right. And this one is about people, and they show it in this kind of do you see the base, do you not see the base format. One baby is looking at, it's like new you looking at old you or something like that. Last two, I think. Why your DNA isn't your destiny. So everything that we've talked about so far in Mendelian genetics is if you have a sequence, an allele, you get an output. If you have a different sequence, a different allele, you get a different output. That output might be nothing if the allele is broken, but it's the sequence that absolutely determines what you're going to get and ultimately what the phenotype will be. But it turns out that there's an area of genetics that we'll touch on later called epigenetics, where the sequence isn't as important as you might think. Okay? You can impose controls on top of DNA sequences such that even if you have two absolutely identical sequences, in one cell it does one thing, and another cell it does another thing. This is kind of not really a Mendelian idea, right, where you have the sequence and you know what you're going to get. The most obvious example of this kind of epigenetic example or phenomenon would be if you think about all the cells in your body. You've got skin cells, brain cells, spleen, liver, all these things. And they're all different types of cells, right? They have very different looks to them, morphologies, they have very different functions, all that. And yet the genome, the sequence of DNA in every one of those things is exactly the same. So there has to have been some sort of epigenetic control placed on top of the genome so that some cells with this genome do one thing, other cells with exactly the same genome do something else. That's an example of epigenetic type of regulation. It's not just the sequence, you have to then regulate on top of it. And the last one is the gene machine, what the CRISPR experiments mean for humanity. So this is a little bit more recent, the idea of CRISPR as a way to go in and specifically target any sequence of DNA that you want for modification. Has been in the news a lot, designer babies, all this type of stuff. So these things are present on the covers of these things. If you go to the supermarket or take a look and see when there's a genetics related thing, because you can understand this. Just fun to look at. Exam on Tuesday. Be here early enough. Be cool. Study hard. I'll see you.